and Life Education presents www.pathshalamasti.com, an educational portal. Here we bring a fast-track course for our students of class 10th. Myself, Neelam Rathore, bio teacher in Vidya Bharti School, is going to discuss Term 1 syllabus of class 10th. In this syllabus, the chapters which are included are life processes, control and coordination, along with practicals. First one, to prepare a temporary mount of the given leaf peel to study stomata. Second, to study that carbon dioxide is produced during respiration. Third, to experimentally prove that light is essential for photosynthesis. Let's start. Life processes. How can we say whether something is alive or not? We can see various activities in different organisms which are actually essential to sustain life. In this chapter, we will discuss all such essential, indispensable processes which sustain life. Before proceeding further, let's enlist these life processes: nutrition, respiration, circulation, or we can say transportation of substances, and excretion. From this section, though not very frequently asked, but one question can be asked. The question is, can you enlist life processes?
Yes, ma'am. As you said, these are nutrition, respiration, transportation, and excretion.
Good. So let's discuss the first life process, that is nutrition. As we have said, that movements in plants, animals are observed, and they are one of the essential life processes. So we can see like flying butterflies, which also need nutrition for their movements because it provides energy. Likewise, the movements are also observed in plants. Say for example seed germination. We can see here that the seedlings are growing, roots and the shoot are forming. That also needs nutrition for plants, which is actually in the form of reserve food material. We can see the upcoming seedlings. In the same way, blood circulation takes place in human beings. The heart is pumping, blood is circulating. That is also an energy consuming process. Then shedding of leaves. At that time, the plants are throwing the waste out, and the reserve food material is utilized by the plants during that particular process in the absence of leaves. So all these life processes which are basically dependent on nutrition. Let's enlist various life processes. As we know, there are many life processes. We can enlist them. Like nutrition is the first life process. In the same way, respiration is another life process. Transportation of substances is one of the essential life process. Likewise, excretion is also another life process. So these are various life processes. Let's take nutrition and its types. Every living creature is active and therefore needs food which provides energy. Various types of nutrition which are observed in different creatures are of two types: autotrophic nutrition and heterotropic nutrition. As we can see here, autotrophic nutrition is shown by all the green plants as well as cyanobacteria, also known as blue-green algae. In the same way, heterotropic nutrition is a broad category. it can be further broadly classified into further subtypes, which are three: saprotrophic nutrition, parasitic nutrition, and holozoic nutrition. Saprotrophic nutrition, as we can see, is normally shown by fungi and bacteria. Likewise, parasitic nutrition, we can see a leech sticking to a human body part shows parasitic mode of nutrition, it is sucking blood. Then holozoic mode of nutrition, that is shown by smaller creatures as well as macro creatures. Here we can see amoeba showing holozoic mode of nutrition. Human beings also show holozoic mode of nutrition. So from this section, now the question comes, can you define nutrition, or what is nutrition? Answer is simple. The process of obtaining food is known as nutrition. Let's start autotrophic nutrition. Here we can see the sunlight is trapped by the chloroplasts found in the leaf. Carbon dioxide enters through stomata. The water, it enters via venous system found in the leaves, and glucose, which is simplest form of carbohydrate, is manufactured. After this process, oxygen, which is produced, we can see here, moves out through the stomata itself. So in this mode of nutrition, living beings prepare their own food with the help of simple inorganic substances like carbon dioxide and water in the presence of sunlight and chlorophyll. This mode of nutrition, as told earlier, is shown by all the green plants and cyanobacteria. Now the question comes that name the mode of nutrition shown by green plants or shown by cyanobacteria, and the answer is autotrophic nutrition. This particular nutrition is also known as photosynthesis. Hence, the question arises now, define photosynthesis. It's a process of manufacturation of food done by plants in the presence of carbon dioxide and water by utilizing sunlight, chlorophyll, and so on. The byproducts are also released during this particular process. Let's have a look what happens during chemical equation of photosynthesis, how we can write it down. 6 molecules of carbon dioxide when react with 12 molecules of water, glucose, simple carbohydrate, is manufactured. 6 molecules of oxygen are released, and 6 molecules of water are also released in the presence of sunlight and chlorophyll. While writing the equation, we should also label glucose, oxygen, and water. Related to this, there are few questions which are asked, like what are the raw materials of photosynthesis? Answer is very simple. Carbon dioxide and water are the raw materials for photosynthesis. Likewise, another question, what are the main products and byproducts of photosynthesis? Simple. Glucose is the main product of photosynthesis. Oxygen and water vapor released out are the byproducts of photosynthesis. Whatever is produced along with the product is actually known as byproduct. It can also be asked that what are the requirements, means the conditions which carry forward photosynthesis. Answer is sunlight and chlorophyll are required for photosynthesis. Photosynthesis occurs in chloroplasts, which are distributed in all the green parts of a plant and are abundant in leaves. Let's see the distribution of chloroplast within the leaf. This is a cross section of a leaf which reveals upper epidermis and lower epidermis. Below to it, some elongated cells which make palisade parenchyma. Below to it, the second layer, which is spongy parenchyma. Collectively, that makes mesophyll tissue within the leaves. Within these cells, chloroplasts are found in the leaves. So these are the actual sites of photosynthesis. Now let's go through what are the main events that occur during photosynthesis. These events are like sunlight is trapped by chlorophyll, which is found in the chloroplasts. This light energy is converted into chemical energy, like we can see glucose is formed, which is a chemical form of energy, and the water which enters is broken down into hydrogen and oxygen. This process also needs sunlight, means breaking of water molecules also need sunlight. Then the carbon dioxide which enters through the stomata is reduced to carbohydrates. Oxygen is released out. Hence, now the question from the section comes that what are the main products and byproducts of photosynthesis? Answer is simple. The main product is glucose, simplest form of carbohydrate, and byproducts are oxygen and water vapor. Now let's discuss photosynthesis in desert plants. Like cactus is one of the desert plant. We can see spines over the cactus found in xeric conditions where water is quite very deficient in the soil, as well as during day the temperature is very high, and all we know that stomata not only help in the uptake of carbon dioxide but also release water vapor. So due to this high temperature during the day, the stomata gets closed. Another plant, euphorbia, we can see on the screen, is also a xerophytic plant. The same happens there. Here also the stomata closes during the daytime due to high temperature. So how photosynthesis occurs in desert plants? Obviously, at night, the moonlight present at night triggers opening of the stomata. Carbon dioxide diffuses in, and hence the manufacturation process, photosynthesis, occurs at night. So photosynthesis in desert plants occur during night. Now the sources of carbon dioxide and water, what are they for the plants? We have been observing and visualizing various types of diagrams. Air is the source for carbon dioxide, and water, obviously it is absorbed by the plants through the roots. So air and soil are the sources of carbon dioxide and water respectively. We have seen that carbon dioxide, oxygen, water vapor, they diffuse in and out through the stomata. What are these and what is their structure, what functions they perform? Let's have a look on the structure of stomata first, and then the rest will carry out later on. Here you can see the structure of an open stomata. The other one is actually a closed stomata. Stomata is this pore, stomatal pore, which is surrounded by two bean-shaped guard cells, normally in dicot plants, and these are surrounded by other epidermal cells. So these cells are also known as the subsidiary cells. Here you can see it is showing the low-lying cells. This reflects the stomata is open. The guard cells are actually modified epidermal cells. In them nucleus is quite very prominent. It can be seen in the microscope. Along with that, we can see round structures. These are chloroplasts. These chloroplasts are found only in the guard cells which are epidermises, modified epidermises. In rest of the epidermises, chloroplasts are missing. Whenever water diffuses out, the guard cells are flaccid and hence the closure occurs. So stomata gets closed. So in this way, this is the structure of the stomata. Now what's the definition? How can we define them? So these are minute pores which are present on the lower surface of the leaf surrounded by bean-shaped guard cells in dicot plants. Now, what's the function of the stomata which are present normally on leaves? They perform specifically two functions. First, that is exchange of oxygen and carbon dioxide occurs. Second, these also do transpiration. Transpiration means release of excess water in the form of water vapor. Now, the question can be asked like name minute pores which are present on the leaf surfaces and what is their function? Answer is quite very simple. These are known as stomata which are present on the leaf surfaces and they perform two functions. In brief, with two words you can write down: function first, gaseous exchange, and function two, transpiration. So in this way, these are the stomata, minute pores present on leaves. Till now, we have been discussing photosynthesis, and all we have understood that during photosynthesis, the plants manufacture starch. Then what about the other food components which are formed in the plants? Say for example, proteins. How the proteins are manufactured in the plants? We have read in previous classes that proteins, they are made up of basically four elements: carbon, hydrogen, oxygen and nitrogen. Hence, plants show absorption and use of nitrogen. Nitrogen is absorbed from the soil in compound state. It is taken up along with the water and it is used in the form of nitrate and nitrites by the plants. They are used in making up proteins. So proteins are manufactured with the help of nitrates and nitrites. During this process, sunlight is not required. Now let's start heterotropic nutrition and its types. In this mode of nutrition, living beings draw their food from other creatures. This food which is taken is in complex form, and it is shown by all the non-green plants, animals, bacteria, fungi, etc. Now let's have a look on types of heterotropic nutrition. It is divisible into three subtypes: saprotrophic nutrition, as you can see here in the diagram, bacteria as well as fungi, they are growing on the food particles, food materials. Second, parasitic nutrition. A long tapeworm is shown in the diagram. It is a parasitic creature. Third, holozoic nutrition. One of the simplest creature is shown, which is amoeba. It is an example of a holozoic creature, draws its food in complex form from the nature. The type 1 heterotropic nutrition is saprotrophic nutrition. In this mode of nutrition, creatures draw their food from dead and decaying organic matter. Examples, as we all know, fungi and bacteria, which are also known as saprotrophs. Here we can see the fungi and bacteria, they are growing on different food articles. On the wood, the fungus is observed. It is found growing and it is decomposing the material. Likewise, in melon we can see bacteria growing. In the same way, in this particular food item, we can see some of the bacteria, it is getting rotten. Mushroom, another example of saprophytic nutrition, is a fungi, and we'll see it growing on the wood. Here you can see beautiful mushroom growing on the wood. Obviously it is a dead and decaying wood, and there we can see these structures on different types of pieces of wood. So mushroom one of the fungi, which is a macro creature basically, shows saprotrophic mode of nutrition. Now let's have a look on parasitic nutrition. In this mode of nutrition, the parasites draw their food from the host and in return harm them. We can see here a parasitic plant, Cuscuta dodder, commonly known as dodder plant, is found clinging around a particular tree. This particular branch of a tree is the host of this Cuscuta. Wherever we can see it is touching, it absorbs food. Another is leech, which also shows parasitic mode of nutrition. The leech, it keeps on finding its prey, attaches to itself and finds the food. Basically, it absorbs blood of other vertebrates. The suckers, with the help of suckers, east, it moves forward. We can see it is contracting and then it is relaxing. It keeps on finding its prey here and there, though it is found in nature, but whenever comes in contact of any vertebrate, it will start absorbing its food. Here we can see how it is moving in search of the prey. The next one is tapeworm, endoparasite of human beings. It's a segmented creature, we can see. It is inside the human intestine, absorbs digested food and in return obviously harms the host. It creates various kinds of diseases in human beings. Hence, Cuscuta dodder, tapeworm, leech are parasites and show parasitic mode of nutrition. Now the question comes, which mode of nutrition is shown by fungi and Cuscuta plant?
Ma'am, in Cuscuta, I think it is saprotropic nutrition and in fungi it is parasitic nutrition.
Perhaps you are not listening it carefully. In Cuscuta, it is parasitic nutrition and in fungi, it is saprophytic nutrition. There are various steps in holozoic nutrition because complex food is consumed. Let's write down these steps. First one is ingestion. Second step, digestion. Third, absorption. Number fourth, assimilation, that is use of food, digested food in the body. And the last one, egestion, that is removal of waste material. So these are the steps in holozoic nutrition: ingestion, digestion, absorption, assimilation and egestion. The first step, ingestion, means intake of food. The food is taken in. In some of the creatures it is swallowed as such, or in others it is chewed and then it is swallowed. As I have said, it is complex food component. Then is digestion. During this process, breakdown of complex food material occur and it converts into simple food components which are soluble in water. Then is absorption. The digested food, which is readily soluble in water, is absorbed and moves to the body, that is via blood or other body fluids. Assimilation, it is utilization of the digested food in various metabolic processes like use of amino acids for the formation of proteins, etc., is known as assimilation. Egestion, the undigested waste which is of no use is thrown out through specific paths in different creatures out of the body. Hence, these are all important steps of holozoic nutrition. Now, let's discuss holozoic nutrition. In this mode of nutrition, creatures depend on solid organic food which undergoes digestion and then is utilized. Hence, in this particular mode of nutrition, there are certain steps which help in the utilization of the food. First, ingestion, then digestion, absorption, assimilation and egestion. Now let's have a look in nutrition in amoeba. It's a unicellular eukaryotic creature found in water. In this particular creature, holozoic nutrition can be very well observed. Here we can see this irregular shaped creature found in water. When it encounters any food particle, then it produces two outgrowths. These two outgrowths are known as pseudopodia. All we know, pseudopodia are not only required for movement, they are also helpful in ingestion. So the food material is enclosed, it is engulfed. The intake of food material through the cell membrane is also known as endocytosis. So the food is taken in the form of a food vacuole. We can see very well in this diagram. This particular food, as I said, is a complex food material, cannot be utilized, hence it needs digestion. So in the vacuole, digestion takes place. This type of digestion is known as intracellular digestion. From the cytoplasm itself, the enzymes keep on diffusing in the vacuole. The food is converted into simple form, which is soluble in water. It comes out, means it diffuses out into the cytoplasm for utilization, and then the leftover waste material, which is still complex, cannot be utilized, is thrown out through the cell membrane. Here we can see the waste particles moving out through the cell membrane. This movement of waste is known as egestion. So all the steps of holozoic nutrition are carried out by the single-celled creature known as amoeba. Let's see the structure of human digestive system. This digestive system is also known as human alimentary canal. Various parts are shown in the diagram. First is mouth, which is also known as buccal cavity. Inside the buccal cavity, tongue is present, teeth are also present. Then buccal cavity leads to a pipe which is known as esophagus. This pipe, it enters via a muscular partition, which is though is not a part of human alimentary canal. It is known as diaphragm. So it leads down in the abdomen. Esophagus opens into a bag-like structure which is known as stomach. At the base of this esophagus, there are sphincter muscles, circular muscles known as the esophageal sphincter. Stomach then leads into the first part of the small intestine, which is known as duodenum, and this is the longest part of the alimentary canal, so it extends down and is surrounded on all the sides by the large intestine. So small intestine starts from here and ends here. It has various subparts also: duodenum, jejunum and ileum. When the stomach leads into the small intestine, here also sphincter muscles are there, which is known as pyloric sphincter. The small intestine then communicates with the large intestine, again with the help of sphincter muscles. Large intestine has some parts. The first part is known as appendix. This appendix then leads into the colon. These are colon, and it's a part of large intestine. Colon communicates with rectum. Rectum opens out of the body through one opening, which is known as anus. Associated glands with the digestive system are liver, which is also known as the largest gland of the body. This liver, whatever it secretes, all we know, that is bile juice, is stored in the gallbladder, where it gets concentrated and then it is released out whenever required. Other associated gland outside of this system is pancreas. So liver and pancreas commonly pour their secretion in the duodenum. Now let's briefly understand the functioning of all these parts. Buccal cavity helps in masticating the food, tongue helps in mixing the saliva with the food. Digestion begins here only. Esophagus carries the food from buccal cavity into the stomach. In stomach, gastric glands are present in the wall of the stomach. these gastric glands release gastric juice. Digestion, which has started from the buccal cavity, continues here, but it is partial. In the stomach, only one component is digested, that is protein, that too partially. Means the gastric juice has hydrochloric acid, mucus, pepsinogen, and another enzyme only found in children, small children, that is renin. Then the next part, small intestine, is actually the site of complete digestion. The digestion for it medium is facilitated, created by the bile juice. Bile juice creates alkaline medium here, just like in stomach acidic medium is created by hydrochloric acid. So here that medium converts into alkaline. Bile juice makes it. Pancreatic juice is active in the alkaline medium, so the enzymes become active. Various enzymes which start digestion in the duodenum are secreted by the pancreatic juice. They are like trypsin, then amylase, lipase, which start the digestion. The walls of the intestine also release intestinal juice, which complete the digestion. And then the digested food is absorbed from the ileum part of the intestine. Then it goes into the large intestine, where already we know the food is solidified, which is waste, and is expelled outside of the body. Now, related to this particular system, some questions are there. For example, like what is the function of hydrochloric acid in the stomach, which is a part of the gastric juice? Hydrochloric acid performs two functions. First, it creates acidic medium in which inactive protein digesting enzyme pepsinogen is converted into active pepsin, so that digestion of proteins can occur. So proteins are converted into peptones. Second, harmful bacteria which are found in the food, if they are there, they are killed by hydrochloric acid. Another question which is related to the digestion and digestive system is that can we name two enzymes or few enzymes which help in digestion, any of them? So we can give answer like the enzyme can be in the buccal cavity. We have read salivary amylase, so one enzyme salivary amylase. Second, in the stomach we have read that is pepsinogen, so we can say pepsin. Or the answer can be pancreatic enzymes like trypsin and lipase. Before starting holozoic nutrition in human beings, let's have a question which we have read recently from the topic, like what is the role of pseudopodia in amoeba?
Ma'am, it helps in ingestion.
Correct. The pseudopodia help in ingesting food material. In the same way, holozoic nutrition is shown in human beings, and for the system the food has to be digested. So here we can see the digestive system of human beings. Various parts are there shown in the diagram. The first is oral cavity. Teeth are present, tongue are there in the oral cavity, tongue is there in the oral cavity. So the food is cut, chewed and masticated. Salivary glands release saliva and partial digestion starts, we can see. That is, only starch is digested in the buccal cavity. Now the food, it passes on, moves into the esophagus, doesn't go into the trachea, we can see epiglottis closing it. Peristalsis movement are clearly seen in the diagram. The food enters into the stomach through esophageal sphincter muscles. The food in the stomach is again digested. Though the digestion is partial, gastric juice, it is made up of mucus, pepsinogen, hydrochloric acid, and small children, renin enzyme is also found in this gastric juice. So gastric juice helps in converting the food into complete paste, as well as digestion of proteins occur in the food. Once the food stays there for approximately 3 hours, and then it is pushed into the next part. So the food, we can say, it moves into the next part of the alimentary canal, that is small intestine, through pyloric sphincter muscles. Duodenum, the first part of the small intestine, then jejunum, ileum, the various parts of the small intestine are there where the food enters. Duodenum is the site for complete digestion of food. The food, it stays there, we can see the movement, and the food is digested completely. For digestion, in the duodenum, certain juices are received. These juices are from the liver, bile juice comes into the duodenum. From the pancreas, pancreatic juice comes. Here we can see there is only one common duct which brings the juices enter into the duodenum through the hepatopancreatic duct, is a site for complete digestion. Bile juice creates alkaline medium for the activation of enzymes in the duodenum. Pancreatic juice starts digestion in the duodenum, and it is actually completed by the intestinal juice which is secreted from the walls of the duodenum. So in this way, in duodenum, digestion, it is taken up by three types of juices: bile juice, pancreatic juice, and intestinal juice. The digested food is then passed on to the next part of the intestine, that is ileum. The inner wall of the ileum, we can see numerous finger-like folds. These are known as villi, which increase the surface area. Look at, these are finger-like folds which are known as villi. Villi increase the surface area enormously, which facilitate rapid absorption of food. If we see the anatomy, the internal structure of villi, then internally they are also manufactured in such a way that facilitate their absorption. Here we can see lacteal and blood capillaries found in the inner part of the villi, which help in the absorption of digested food. The capillary network, that is basically blood is flowing there, all the food is absorbed through that, except the fat molecules. Digested fats are absorbed by the lacteal. Lacteal are actually lymphatic capillaries. So in this way, the villi help in absorption. Hence the inner part of the intestine, that is ileum, is designed in such a way that it helps in the absorption of food. This absorbed food then passes on into the blood stream. We can see the movement of the food, and we are seeing the flowing RBCs also. This food goes to each and every part of the body. Now we can see the passage of food. It enters into the large intestine. Obviously this food is undigested food. Through the ileocecal valve, the food enters here. Wherever valves are there, they prevent the backflow of the food. So this valve is ileocecal valve, and hence the food comes into the large intestine. These parts are colon. So the colon in the large intestine helps absorption of water, manufacturing of vitamins, specifically vitamin B12, production of mucus to you can say remove friction, formation of feces which are expelled out. The feces, they come here, and then through the anus they are removed outside of the body. In this way, large intestine helps in excreting the undigested waste. So now the question comes that which food component is digested in our buccal cavity?
It's starch.
Correct. The starch is digested in buccal cavity. Can you tell me that starch, it is converted into which simplest form in the buccal cavity?
It's glucose.
No, it's not glucose. It is maltose, which is a disaccharide made up of 2 glucose units, means it also needs further digestion. Hence, digestion begins right from the buccal cavity. As we have understood various steps in holozoic nutrition, digestion is one of the important steps during which the food is converted. Actually, what are the end products of digestion? Whatever we eat, what is formed? Let's have a look, because it is an important part. Normally it is asked in examination. So let's take the first food component, that is starch. A complex food component is converted into simple food component. Let's see what happens. It converts into glucose. This is one of the simplest component, readily soluble in water, quickly absorbed by the blood, and goes to each and every body cell. It's actually a concrete reaction various enzymes play a role in this conversion. Let's write it down. They are pancreatic enzymes as well as intestinal enzymes, and the beginning takes place with the help of salivary enzymes. So we can write down all of them together, that they convert collectively starch into glucose. The next food component is protein. All the proteins are converted into amino acids, readily soluble again, and move into the blood. Proteins are converted into amino acids. These are complex, these are simple, by the help of various enzymes. These are pancreatic enzymes, gastric enzymes, and intestinal enzymes. Likewise, fats are also converted into simple food components. The simplest form of fats are fatty acids along with glycerol. This conversion takes place by means of pancreatic and intestinal lipases. So this way, these are the end products of all the complex food components which happens during digestion. Here the question comes that what is the role of enzymes? So enzymes are biocatalysts which convert complex food components into simple food components. Right now we have discussed the human digestive system. After digestion, the food is absorbed from the ileum. How the ileum is designed to facilitate rapid absorption of food? Let's see the internal details. The wall of the ileum has numerous finger-like folds. Here we can see these are finger-like folds, which are known as villi. They are actually the epithelial lining that makes the villi, and this is the outermost layer which is towards the lumen of the ileum, which is known as mucosa. Hence, here the finger-like folds are present throughout the ileum wall. This we can say that if we stretch it, the surface area will increase enormously. Inside, the connective tissue is present, in which it is supplied by lymphatic vessels. It has arteries, veins to supply blood as well as to carry it away. If we see the internal structure of one villi, which is actually shown here, the singular form of villi is known as villus. So this is one villus. Here we can see internally it has numerous blood capillaries. We can say this is blood capillary, oxygenated blood is shown and deoxygenated blood is shown also, and then lymphatic vessels are also there, and the outer layer is known as the epithelium. So when the food enters in the ileum, these villi absorb the food which goes into the blood capillaries. Fat, it enters into the lymphatic vessels, and from the ileum it diffuses into the blood. From there it is circulated to each and every part of the body. Right now we have read about holozoic nutrition in human beings. So now the question arises that what is emulsification of fats? Through the mechanism of digestion, I have told you that the starch digests in the buccal cavity, proteins are digested in the stomach, but nowhere fats were discussed. Means fats, their digestion begins at the end. The first step is, what is emulsification of fats? So conversion of big fat globules into smaller ones is known as emulsification of fats. It is done by bile juice to facilitate smooth and easy digestion of fats. When the food enters large intestine, then what happens there? Is it a useful part of the alimentary canal, or it is just like storing the waste? Let's have a look. So the functions of large intestine are displayed here. We can go through them. It stores undigested food. Then absorption of water from the stored food material takes place, and the food, it converts into feces, solidifies. Then this food is pushed into the last part of the large intestine, which is rectum. Through there is one opening, anus, which expels the wastes out. Hence, it is not only storage of waste material, it is also absorption of water and expulsion of feces. Let's start with the next life process, which is respiration. All the creatures breathe, or we can say respire, continuously. Can you think why? Why do they respire continuously? The answer is very simple. To inhale, to get oxygen, and that produces energy in the body. It also produces carbon dioxide that is released out. So during the process of respiration, breakdown of glucose occurs, energy is liberated, which is utilized in various different metabolic processes. This respiration is also known as a catabolic process, because the glucose is broken down into simple inorganic substances, carbon dioxide and water. We are discussing respiration. Like, take example of running children. We can see the children are running. The respiration rate, or we can say the breathing rate, is faster whenever we are more active physically. So here the playing children with football, obviously they are respiring much fast. So this is one mode of taking more oxygen at that time. Let's take example of resting children. If they are sitting, not physically very active, at that time the respiration rate is comparatively slow. We can see the dog is also resting here. So all the creatures, when they are resting, they take less oxygen. This depends on the liberation of energy, how much energy is required and how much oxygen is required. So now the question comes, define respiration.
Ma'am, it is a catabolic process during which glucose is broken down and energy is released.
Good. During this process, obviously glucose is the food material which is broken down and energy is liberated, which is utilized further. Respiration in human beings or in whatever living creatures completes in two steps. The first is breathing, second internal respiration. Breathing, simply intake of oxygen rich air and release of carbon dioxide rich air. So it is a physical process during which oxygen rich air enters in our respiratory system, in the lungs, and carbon dioxide rich air is thrown out. Being a physical process, it is actually energy consuming process. So energy is consumed during breathing. On the other hand, internal respiration, it occurs inside the lungs. So inhaled oxygen diffuses into the blood and that is circulated into the body and goes to each and every body cell up to cell and tissue level. That particular oxygen which is circulated, which is diffused out of the lungs, it is utilized in oxidizing the food material, that is glucose, and energy is released. So in this way we can also find out the differences between breathing and internal respiration. Types of respiration. As we know, respiration is a continuous process. It occurs in living beings. It is basically of two types dependent on the presence or absence of oxygen. we can understand it by making a flow chart. We can write down respiration first, divisible into two categories. Anaerobic respiration, that is the first type. Second is aerobic respiration. During anaerobic respiration, oxygen is not used. When oxygen is not used, incomplete breakdown of food particles take place. While in aerobic respiration, oxygen is used. This use of oxygen helps in complete breakdown of food particles. Respiration is basically of two types depending on the presence or absence of oxygen, whether oxygen is utilized or not. If oxygen is not utilized, that is known as anaerobic respiration. If oxygen is utilized in the breakdown of food molecules, then it is known as aerobic respiration. So we can make out the differences between aerobic and anaerobic respiration. Along with that, we can also understand the definitions, the end products, and what happens in which creatures these respirations occur. During anaerobic respiration, as said, oxygen is not utilized at all for the breakdown of food molecules. Hence, incomplete breakdown of food occurs. On the other hand, in aerobic respiration, oxygen is fully required. Hence, complete breakdown of food takes place and the end products are basically non-toxic. In the same way, when incomplete breakdown of food occurs, less amount of energy is released during anaerobic respiration, that is only 2 ATPs of molecules. On the other hand, in aerobic respiration, due to complete breakdown, lots amount of energy is released, approximately 38 ATPs from one glucose molecule. The end products during anaerobic respiration, which are formed after the breakdown of glucose, are slightly toxic. So the toxic end products are there, which we can take example lactic acid or ethanol. They produces toxic effects in the creatures. On the other hand, non-toxic end products are produced during aerobic respiration, carbon dioxide and water, which are thrown out quickly from the body. In anaerobic respiration, pyruvic acid, which is produced after the breakdown of glucose, that is further fermented, and the fermentation, end products of the fermentation, they can be either lactic acid or ethanol. The whole of the process occurs in the cytoplasm itself. So anaerobic respiration occurs in the cytoplasm. Aerobic respiration means after formation of pyruvic acid, this is actually similarity. The first step is similar. Glucose forms pyruvic acid. Then after, this pyruvic acid enters into the cell mitochondria. There it is broken down completely. Hence, aerobic respiration takes place in the cell cytoplasm and then in the mitochondria it completes. So this way we can differentiate very well between aerobic and anaerobic respiration. Now the question comes that what happens to food during aerobic respiration?
Ma'am, complete breakdown of food occurs in the presence of oxygen and lots of energy is released.
Good. This energy which is released is approximately 38 molecules of ATPs, and carbon dioxide which is also released, that is thrown out. So aerobic respiration helps in providing enough energy. As we know, glucose is formed during digestion. What happens to this glucose during respiration? Actually, breakdown of glucose occurs during respiration. Let's have a look how this process takes place. The glucose is converted into pyruvic acid. This conversion of glucose into pyruvic acid doesn't need oxygen. So oxygen is not used. Where does this process takes place where oxygen is not required? In a cell. So we can write it down. In the cytoplasm of the cell. After that, the pyruvic acid is further broken down into simpler components. This breakdown of pyruvic acid occurs inside the mitochondria. And for this breakdown, oxygen is required, means the second step needs oxygen. So oxygen is used. Now this pyruvic acid further completes and completely breaks down and forms 6 molecules of carbon dioxide, 6 molecules of water, which are normally released out, along with 38 molecules of ATPs. These are energy currency of the body, which we can also write it down that it is energy. So one molecule of glucose completely breaks down into 6 molecules of carbon dioxide, 6 molecules of water. Energy is liberated in the form of 38 ATPs, which is utilized in various metabolic processes. Right now we have read about breakdown of glucose molecule. It's an important part of the chapter that we should know what happens to glucose. So the question from this particular part can be that what happens to glucose molecule during aerobic respiration. Let's have a look. The answer can be written this way: glucose converts into pyruvic acid. During this particular process, oxygen is not utilized. So this is anaerobic step of aerobic respiration. This is the special quality of this. It occurs in cytoplasm. Overall, this conversion is known as glycolysis, conversion of glucose into pyruvic acid. Then this pyruvic acid breaks down into carbon dioxide, 6 molecules, then 6 molecules of water, along with energy, which is released in the form of 38 ATPs. This is energy. This particular process takes place in the mitochondria. So we should mention mitochondria. Oxygen is utilized in this particular process. So we should mention oxygen is used. This overall process is known as Krebs cycle, which completes after ETC, electron transport chain, within the mitochondria. So during aerobic respiration, glucose converts into carbon dioxide, water and energy, which is 38 ATPs of molecule. So in this way we can remember this equation. Right now we have read what happens to glucose during aerobic respiration. Now let's take another example what happens to glucose molecule during anaerobic respiration in yeast. Because yeast performs anaerobic respiration, it needs glucose for getting energy. So what happens to it? Glucose in yeast is broken down into pyruvic acid just like bigger creatures. For this process, again, oxygen is not required. This process, it occurs in the cytoplasm of the cell and is again known as glycolysis. Further, this pyruvic acid is converted, rather we can say it is fermented into carbon dioxide molecules, ethanol, which is also a toxic component. Rather we can write it down first, the main product. It means we can say ethanol, then carbon dioxide, then energy. This energy is very less, so we should mention 2 molecules of ATPs. Now during this process, what are the requirements? Again, oxygen is not required in this process. And where does it occur? In the cytoplasm of the cell itself. What is the term used for this conversion? Fermentation. So here we can see glucose in yeast converts finally into ethanol, carbon dioxide and energy. Very less amount of energy is released, that is only 2 ATPs of energy, and the whole process takes place in the cytoplasm and is also known as fermentation. We have read that glucose breaks down anaerobically in yeast. Does it occur in complex creatures also? Let's have a look. The question is, what happens to glucose molecule during vigorous physical exercise in man? Yes, means we have to write down what actually is formed by this glucose. Glucose, the substrate for oxidation. As we all know, it converts into pyruvic acid. For this conversion, oxygen is not required. This process is known as glycolysis and it occurs in the cell cytoplasm. After that, this pyruvic acid it remains, means in our muscles, glucose which is already present, once pyruvic acid is formed during vigorous exercise, this pyruvic acid is converted into lactic acid. Along with lactic acid, carbon dioxide is released and energy also. This energy is again very less, means only 2 ATPs of energy. Then what's the difference? Means this pyruvic acid is converted into lactic acid, obviously in the absence of oxygen, means oxygen again is not utilized. And this is actually known as fermentation, which occurs in human beings, and it occurs in the cytoplasm of the cell. Means in whole of the process, mitochondria is not taking place. Glucose converts into lactic acid, energy is released which is less in amount, and this takes place so that continuous energy is released in the body, because during physical exercise lots of energy is consumed. Oxygen deficiency may occur. So to fulfill that, this process takes place. Let's study the respiratory system in human beings. What are its parts and in what way they are arranged. The human respiratory system, as shown in the chart, has following parts. First, the nasal passage, means nose leads into nasal passage. This nasal passage here comes and communicates with the pharynx. We can also see the mouth cavity is written. So mouth cavity and nasal passage, they communicate with the pharynx. So this is a common communication point or the junction point. From here it leads into the trachea via the voice box, which is known as larynx. It is placed above the trachea. Then the trachea, it descends down and it branches into two parts. Here we can see that there are ring-like structures. These rings are made up of cartilages. They are not actually circular rings. They are C-shaped rings which prevent the collapsing of trachea in the absence of air. So there is a continuous passage for the movement of air. In the thoracic cavity, trachea terminates into two branches. These are known as bronchi. It is written bronchi. Right hand side and left hand side bronchi are there. Inside the lungs they enter and further branching occurs. These are finer branches comparatively and are known as bronchioles. The bronchioles then end into alveolar sacs. So alveoli are present within the lungs. Here a detailed structure is shown of alveolus. These are many alveoli within the lungs. Millions of alveoli are present. So these increase the surface area of the respiratory system. This upper part is actually known as the thoracic cavity, which is supported by ribs. Ribs protect the lungs which are present internal to it. And from the lower abdomen, by means of a circular diaphragm, they are separated. It is a muscular partition known as diaphragm. So we have a pair of lungs in the respiratory system. So in brief, if we go through, that is nose, nasal passage, then larynx, trachea, lungs, inside the lungs, the primary bronchi, bronchioles and alveolar sacs. So these are the parts of respiratory system. Now let's have a look at what are the important features of respiratory system. First of all, in our respiratory system, the pharynx is the point where the buccal cavity and the nasal cavity communicates. And at that particular point, glottis is there. So this slit-like opening at the base of the trachea is normally covered by a flap-like structure known as epiglottis while swallowing anything or while drinking water. So in this way, that is prevented. Then trachea, the other feature is trachea is supported by C-shaped cartilaginous rings to maintain a continuous passage for inward and outward movement of gases. Third, millions of alveoli which are present inside the lungs, they increase the respiratory surface area for efficient absorption of oxygen and diffusion of carbon dioxide back. Now let's understand the mechanism of breathing. Breathing, it completes in two steps, inhalation and exhalation. The human respiratory system consists of a pair of lungs which are roughly cone-shaped. We can see here they are filled in the chest cavity. The rib cage protects the lungs. Trachea, which is also known as windpipe, leads into the lungs. A dome-shaped structure is there, which is diaphragm. It is actually a muscular partition which separates the thoracic cavity or the chest cavity from the abdomen. The functional units of respiratory system or the lungs are there, which are known as alveoli. So the respiratory system, it helps in inhalation as well as exhalation. So during the first step, which is known as inhalation, let's see what happens. The air it enters into the trachea, reaches into the lungs. We can see here the lungs expand. Why? Because the thoracic cavity, it has also expanded. So it provides a space for the lungs to expand on all the sides. On the lower side, diaphragm, which is a muscular partition, it moves down. Hence the thoracic cavity expands downwards also. Due to expansion of lungs, the air pressure inside the lungs decreases in comparison to outer atmosphere. Hence the air is drawn inside. The next step will be like exchange of gases will take place quickly through the alveoli, and then exchange of gases, during that carbon dioxide will come back into the lungs. Now it has to be exhaled out. Hence during that particular process, we can see here exchange of gases is taking place in the alveoli. It takes a little bit some time, and then the carbon dioxide will move out. During that movement, again some of the steps will be followed. The exhaled air, when it goes out, how it is possible? Because the diaphragm comes back to its original position, that it becomes dome shaped. The ribs also come back to their original position. Due to that, air pressure inside the lungs increases in comparison to outer atmosphere, and then the air, it moves out, which is carbon dioxide rich. So it is basically the costal muscles between the ribs and the diaphragm which helps in inhalation as well as exhalation of gases. Thus the mechanism of breathing is over. Right now we have seen mechanism of breathing in human beings. What happens to that gas, oxygen rich gas, which enters in our lungs? Let's have a look. That gas, it enters the alveoli. Oxygen rich gas enters in the alveoli, and then this, where we can see the deoxygenated blood which is coming from the heart into the alveoli. Alveolar walls are supported with blood capillaries. Numerous blood capillaries are there, and then these arrows are depicting the movement of gases in and out. So basically the inward moving gas is deoxygenated, the inward moving blood is deoxygenated, carbon dioxide rich, and the outward moving blood is oxygenated, means in the alveolar walls the blood capillaries help in picking up oxygen and moving back carbon dioxide into the alveolar space. That carbon dioxide is taken up and it moves out. The oxygenated blood, it moves via various pulmonary vanules which make pulmonary veins back into the heart, from where it is circulated within the body. Right now we have seen that oxygen is picked up from the alveoli of the lungs. What happens to this oxygen? Where does it go? Let's have a look. The oxygenated blood, which is oxygen rich blood, via heart goes to various body parts, and exchange of gases take place in the same manner as it has happened in lungs, in tissues or cells also. The first step is there, that the oxygen rich blood which goes to various body cells via heart, there the oxygen diffuses within the tissues and cells. Carbon dioxide comes back into the blood. What's the use of this particular oxygen? That is used for releasing energy. The carbon dioxide which is picked up, again it goes into the veins, and all those veins carry the blood back to the heart. So basically, in exchange of gases in tissues and cells, first is that oxygen rich blood goes to the tissues so that the glucose can be broken down, and due to that the oxygen is continuously used up, hence its amount decreases. So oxygen is less in the tissues. This oxygen diffuses out from the blood into the tissues. Carbon dioxide which is already more comes back into the blood, and then oxygen and carbon dioxide are transported likewise within the body. So we have been discussing till now breathing, internal respiration, exchange of gases, and so on. Many a times you might have observed that whenever we are doing something, we are moving from a stinking street, we hold our breath. How come it is possible? Is respiration continuing at the time? The answer is yes, it is continuing. How it is possible? There is some amount of air which is trapped in the alveoli, trachea, and in the bronchioles, the branching system of the lungs. That air which is trapped is known as residual volume. This volume of air cannot be exhaled out even after forceful exhalation. So this residual volume helps in continuing respiration even if we stop breathing. So now the question can be asked, that is respiration a continuous process even if we stop breathing?
Yes, ma'am. The residual volume of air present in lungs helps in continuous respiration.
Correct. It helps in continuous respiration. Now, from this text, whatever we have read in respiration, one more question can be asked, that does anaerobic respiration occur in multicellular creatures like human beings?
Yes, ma'am.
How it is possible?
Ma'am, during physical exercise, anaerobic respiration occurs in human beings. At that time, glucose converts into lactic acid and energy is released.
Correct. So that during vigorous exercise, the requirement of oxygen is more, and maybe we have a capacity of supplying oxygen up to a limit. So to maintain a continuous supply of energy, anaerobically glucose is broken down, lactic acid is produced, which is later on metabolized in the body. So henceforth, respiration, the life process, is one of the essential processes to sustain life. As we know, all the living creatures respire and they thrive in different habitat. Some are found on the terrestrial organisms, some are found in water, which are known as aquatic organisms. Is there any difference in their breathing? So let's have a look to understand the difference between respiration in terrestrial organisms and respiration in aquatic organisms. Terrestrial organisms, there the respiration rate is slow. Why? Due to higher percentage of oxygen in the air. But many a times we have observed fish in aquaria or in natural habitats, the breathing rate is faster. Why so? Because the oxygen in water is fairly low. So they have to fulfill the oxygen requirement. For that, they have to respire faster. So this is one of the difference. Another, the terrestrial organisms have lungs, trachea, or in insects, the tracheal system, which draw oxygen from air. But in aquatic organisms, the respiratory organs are gills, which extract dissolved oxygen from the water. So in this way, these are some basic differences between terrestrial organisms and aquatic organisms related to respiration. Now the question can be asked, that why do aquatic organisms breathe at a faster rate? The answer is very simple. Amount of oxygen is fairly low in water, hence to fulfill the energy requirement they have to draw more oxygen, and for that they have to respire faster. Hence in fish and other aquatic creatures, breathing is faster. As we have said, respiration occurs in all the living beings, and we all know plants are also living beings, hence they also respire. We have also understood that when we are moving fast, we are doing vigorous exercise, we take in more oxygen, respiration rate is higher, but when we are resting, respiration rate is slow. Here the question comes basically that plants are static beings. They are static, they are found at one place, they are not moving here and there. So it's very obvious that in them respiration is always slow. So in plants, What are the features that help in respiration, the structures that help in respiration, what type of respiration rate is there? It's their respiration in plants, like they're starting beings, so they need always very less amount of oxygen. It is less oxygen consuming. In plants, there is no respiratory system just like human beings and other insects or bigger creatures. Means the movement of oxygen is less, up to few cells. So this makes very clear that there are different structures which help in the intake of oxygen and its diffusion to some of the adjoining cells. And we also know that respiration is a continuous process, means plants keep on respiring just like animals continuously day and night. And it is just like human beings, they take oxygen in and release carbon dioxide out. Normally a confusion is there that which gas is taken by the plants during respiration. So that is oxygen. Always they take oxygen and release carbon dioxide. We were talking about various structures which help in respiration in plants in the absence of respiratory system in plants. So these structures are root hair found in roots, stomata which are found, all we know, all over the leaves, and lenticels which are there on woody branches, old roots, and they are also small openings. Now the question comes that name various structures which help the plants in respiration.
Ma'am, it's root hair and stomata.
Correct. You have given two points, that is root hair and stomata. Lenticels are left aside. Lenticels are also just like stomata. They are small openings which help in the respiration of older plant parts.
Enlive Education presents www.pathshalamasti.com. I am Mrs. Tanuja Chakravarti, PGT Biology teacher of St. Gregorius School, Dwarka. I welcome all my dear students who have opted for Rachna Sagar Crash Course of Science to my lecture room. I hope our journey together would be meaningful, interactive, joyful and helpful for all of you. Now, let's enter into the vivid world of biology. I am here to take up the Term 1 chapter, Life Processes. The topic with which I am going to begin is transportation. Let's begin with the chapter, Life Processes, topic transportation. As I have told you, our session has to be interactive, hence I would like to begin with a question. What is transportation with respect to living organisms?
It is a life process in which a substance absorbed or synthesized in one part of the body is moved to the other parts of the body.
Very good. Now let's study transportation in the human body. The main medium of transport in the human body is blood. Blood helps in transporting food, oxygen, wastes, salts and hormones. Blood is a fluid connective tissue comprising of a matrix called plasma, in which three kinds of blood cells, namely red blood cells, white blood cells and blood platelets remain suspended. Can you name the constituents of the human transport system?
No, ma'am.
Fine, I'll tell you. The human transport system comprises of a highly muscular pumping organ called the heart, which is interconnected with a network of blood vessels forming a closed circulatory system. The beautiful animation gave us the image. I'll ask you the next question now. Can you name the different kinds of blood vessels which are present within your body?
Yes, ma'am. The blood vessels are of three types: arteries, veins and capillaries.
Now let's study the structure and functions of the three types of blood vessels. Let's have a look, children. Always remember, the blood vessel which carries blood towards the heart, it's known as the vein. The blood vessel which carries blood out of the heart, that is known as the artery. And both the arteries and the veins, they end up in a very thin and minute network which is known as the capillary region or the capillary network. Now let's study the differentiating features between the three types of blood vessels. First, arteries. The arteries carry oxygenated blood from the heart to the different body organs. The veins, they carry deoxygenated blood from the body organs back to the heart, and the arteries and veins, they end up in a very thin and minute network which forms the capillary region. Next, the arteries, they have thick and elastic walls because blood at a very high pressure flows through the arteries, whereas veins, they do not have thick walls because blood is no longer under pressure while it is flowing through the vein. The capillaries, they have only walls which are 1 cell thick. Next children, let's have a look. The arteries, they do not have valves in them because blood flows at a very high pressure through them. The veins, they have valves in them because it prevents the backflow of blood within the veins. The exchange of materials between the blood and the surrounding tissues takes place at the capillary region. Now I'll ask you a question. Can you tell me how many chambers are there in your heart?
Yes, ma'am. Our heart has 4 chambers.
Good. Now let's have a look at this beautiful animation that we are having. The human heart is 4-chambered. It is highly muscular and it is as big as a fist. It is located within our thoracic cavity. Being 4-chambered, it entirely prevents the mixing of deoxygenated and oxygenated blood. Let's have a look. The right side of the heart entirely deals with deoxygenated or impure blood, whereas the left side of the heart entirely deals with pure or oxygenated blood. Next, one thing you always need to remember. Whenever you draw and label a diagram of your human heart, please remember the right side of yours should be labeled as the left side of the heart, and your left hand side should be labeled as the right side of the heart because it's present within your body in this location. Now let's move on further. The upper chambers of the heart, they are known as the atria or the receiving chambers. The lower chambers of the heart, they are known as the ventricles or the sending chambers. Now let's have a look at the major blood vessels which enter and leave the heart. This is our right atrium. It receives deoxygenated blood coming from the major blood vessels called the vena cava. This is our left atrium. It receives pure oxygenated blood coming from the lungs which is entering the heart through the pulmonary vein. Let's come to the ventricles. This is our right ventricle which sends dirty blood out through the pulmonary artery, and this is the left ventricle which sends the pure oxygenated blood out through the main artery called the aorta. There are a couple of valves also present within your heart. Let's locate them. Right at the juncture of the atria and the ventricles, a pair of valves are present which are known as the cuspid valves, whereas the place from where the pulmonary artery and the aorta leave the heart, right at that point, children have a look. Here a pair of valves are present. These are known as the semilunar valves. The ventricles have much thicker and muscular walls than the atria because the ventricles are sending chambers and they have to pump the blood out and send it to the different body organs. Relatively, the walls of the atria are much thinner as compared to the ventricles. Children, this was our discussion about the structure of the human heart. Now, let's go over to the working of the human heart. As we have already discussed about the 4 chambers of the heart, now let's see how they work to pump the blood out of the heart. Initially, all the 4 chambers of the heart, they are in a relaxed state. The atria, they receive blood from the vena cava and from the pulmonary vein. Instantly, both the atria contract. As soon as they contract, the blood gushes from the respective atria into the respective ventricles. Please remember, this is the point where the atria relax. Momentarily, both the ventricles, they contract. As soon as they contract, the pressure of the blood in the contracting ventricles first forces the 2 cuspid valves to close. This is one cuspid valve, this is the other cuspid valve. First and foremost, these 2 valves close. What will happen to the blood? The blood will try to gush out. As a result, the high pressure of the blood allows the semilunar valves to open. As soon as the 2 semilunar valves open, blood starts gushing out from the right ventricle and moves out through the pulmonary artery and is carried to the lungs. On the other hand, blood gushes out from the left ventricle and it starts moving out through the aorta. Instantly, the ventricles relax. The atria were already relaxing. As a result, it's the beginning of the next cardiac cycle. Now let's recapitulate the working of the human heart. The entire structure and working that we entirely discussed, let's have a look with a much clearer picture. See children, this is the right side of the heart, this is the left side of the heart. The right side, as I told you, deals with deoxygenated or dirty blood. You can see the right atria and the right ventricle, they have received deoxygenated blood. From this right ventricle, the blood is sent out to the lungs for purification by the pulmonary artery. The right side deals entirely with dirty blood. But see children, the left side deals with pure oxygenated blood. Have a look, all the 4 valves were shown to you. This is the cardiac cycle on the making. The atria are contracting, next the ventricles are contracting. It's a simultaneous affair. Can you tell me how many chambers are there in the heart of a fish and that of an amphibian?
Ma'am, fish has 2 and amphibians have 3 chambers in the heart.
Okay, let's have a look now. We have here a picture. Here we go. Children, this is a comparative diagram showing the number of chambers in the heart of a fish, of an amphibian, and this one shows that of a mammal. Now, one thing we need to remember. The number of chambers present within the heart of an animal decides whether the animal would be warm-blooded or cold-blooded. Why? Because when the separation is there in the heart chambers, it entirely prevents the mixing of oxygenated and deoxygenated blood. Hence, it also affects the oxygen carrying capacity of the blood. Keeping that in our mind, here we have a look. This shows the 2-chambered heart seen in a fish. From the heart, the blood is pumped to the gills. There the blood gets oxygenated. From there it is distributed to the different parts of the body, and from there it once again comes back to the heart. So here we see the blood moves to the heart only once. So we say fishes show single circulation in their heart. Next, here we have the heart of an amphibian or of a reptile. Here we see the heart has 3 chambers, 2 upper chambers called the atria, and there is only 1 lower chamber called the ventricle. Hence what happens? When the heart pumps the blood out, it pumps out mixed blood. Hence the oxygen carrying capacity of the blood is towards the lower side. So these animals are also known as cold-blooded animals. They cannot maintain or regulate their body temperature constantly. Now children, this is your and mine heart. It is 4-chambered. What happens here? Entirely prevention of mixing of oxygenated and deoxygenated blood is taking place. As a result, blood's oxygen carrying capacity is very high. Hence, being warm-blooded, our energy demands are very high. We need to regulate and monitor our body temperature constantly, which can be done and sufficed because our heart has clearly 4 chambers. Based on our discussion, let me ask you a question. Fishes, their heart showed single circulation. Then, what kind of circulation do you expect to see in your heart?
I think it's double circulation.
Oh, that's good. Let me explain what is double circulation. As the blood moves through the heart twice before it is distributed to the different parts of the body, hence we call it double circulation. Blood once enters the heart, from there it goes to the lungs for purification, from there once again it gets back to the heart and again sent out through the aorta. Hence, in a 4-chambered heart, we find double circulation taking place. Now, let me ask you a question. If there is a leakage in the entire circulatory network of your body, what's going to happen? Can you try out the answer?
No,
Ma'am, it's
ma'am.
difficult.
Okay, children, I'll tell you. If you get a cut or wound, what happens? Blood starts oozing out from there. But after some time, did you notice a clot forms above it? Yes. That process is known as blood clotting, and at the beginning when we studied about the 3 kinds of blood vessels, I hope you remember we named blood platelets. Those cells, the blood platelets come to the rescue and they help in the process of blood clotting and stop the point of injury. If the leakage continues, what's going to happen? It will affect the efficiency of our transport system, plus it will also affect the blood pressure of the flowing blood within our body. The pressure is going to fall. Now let's study what is lymph. Here I have a picture showing the capillary region. Now, as you know children, the artery ends up in the arterial capillary and the vein also ends up into the venous capillary. That capillary bed has been shown here. This is the artery narrowing down into the arteriole. The arteriole ultimately is breaking up into the arterial capillaries. On the other hand, you can see this is the venule, which will actually lead to the vein, and these are the venous capillaries. You need to know the arterial capillary and the venous capillaries, they are present end to end like this. Now, the walls of the capillaries are actually very thin and they even have minute openings present on their surface. What happens is, when blood flows through the arteriole and enters into the arterial capillary, a very high pressure is created because blood is flowing through very narrow channels. Due to this, the plasma part of the blood, which is now called tissue fluid, seeps out from the tiny openings of the capillaries. Children, you can see very minute arrows have been drawn here. The plasma part of the blood that is seeping out through the capillary zone, and it bathes the tissues which are present in the capillary bed. All the yellow colored cells which you can see children, they are the tissues present in this region. What happens? The tissue fluid brings with it oxygen and nutritious substances which are picked up by these cells. After doing this job, the tissues, they absorb the nutrition and the oxygen, whereas they give out the wastes and the carbon dioxide to the tissue fluid. After doing this job, most of the tissue fluid laden with wastes and CO2 now enters back into the venous capillaries. The arrows are now slowly entering into the blue channels, that is the venous capillary. Most of the tissue fluid, almost 90% of the tissue fluid, seeps back into the venous capillary and is carried away with the vein. But some part of this tissue fluid, which carries larger molecules of fats and proteins, which cannot enter back into the venous capillary, now has to enter into another system of ducts present within the body, which have been shown in green children. You can see a network of green tubes in between the cells. The part of it which carries bigger molecules, they enter into another network within the body, which is known as the lymphatic network or the lymphatic system. And now this tissue fluid gets its name as lymph. So this lymph once again gets drained back into our main circulatory stream. So this was a study about what is lymph and how is it formed. Here we have a picture which shows the entire lymphatic system of your body. Now let's focus on the functions of lymph. The first function, it helps digested and absorbed fats from the intestine to be carried away for assimilation. Next, it helps in draining the extracellular fluid or lymph back into the blood. After completing the discussion of the human transportation, now let's get into transport in plants. But children, I would like to begin with some questions first. Can you name the two complex permanent tissues found in plants that you have already studied in class 9?
Yes, ma'am. Xylem and phloem.
Oh, good. Can you tell me their functions?
Yes, ma'am. Xylem helps in transporting water and minerals, and phloem helps in transporting food.
Good. Yes, one more question here. Can you name the components of the xylem tissue?
Yes, ma'am. Components of xylems are vessels, xylem parenchyma and xylem fibers.
Okay. One you missed. Never mind. When we take up the discussion, we'll have a detailed account of it. I am actually testing your previous knowledge, what you have done in 9th. Can anybody tell me the components of the phloem tissue?
Components of phloem are sieve tube cells, companion cells, phloem parenchyma and phloem fibers.
Very good. Now let's study the differences between xylem and phloem tissue. First, have a look at the animation. Children, this is a xylem vessel. We find it helps in transporting water and minerals upwards. Whereas, here you find this is a diagram of the phloem vessel. Here you can see arrows moving both up and down. Now let's refer. Xylem, it helps in transporting water and minerals from the roots to the upper parts of the plant body. Whereas, for phloem, it helps in transporting food from the leaves to the rest of the parts of the plant body. Next, transport through xylem is unidirectional, whereas transport through phloem is bidirectional. Can you define transpiration, children?
No, ma'am.
Okay, I'll tell you. It's the process of throwing out excess water from the plant body in the form of water vapor through the tiny openings present within the leaves called the stomata. Now let's check what is the significance of this process of transpiration. First and foremost, it helps the plant to lose the excess water present within its body. It also helps in regulating the temperature of the plant body by bringing about a cooling effect. Thirdly, it helps in transporting water and minerals within the plant body through the xylem vessel. Now let's study the mechanism of transport of water through the xylem tissue. First, children, the root hair cells, they take up water actively by the process of osmosis. That's the first step. These water molecules slowly move through the different tissues and ultimately it enters into the xylem tissue. Within the xylem, the water molecules form a continuous water column which is slowly pulled up against gravity by the process of transpiration pull. Here we have an enlarged diagram showing the structure of the water molecules, how they pass within the xylem capillary. These water molecules, they have formed a continuous column and this column is pulled out above very slowly by the process of transpiration pull. Here you can have a look, children. From the leaves, water is continuously being lost by transpiration. As a result, the entire column starts moving up. This is how water is transported through the xylem tissue. During daytime, it is transpiration pull which helps in driving the water molecules up the xylem tissue. And during night time, there is another pressure acting called the root pressure which helps in taking water up through the xylem tissue. Now let's discuss the mechanism of transport of food through the phloem tissue. The process of transport of food through phloem is defined as translocation. Phloem helps in transporting food, amino acids and many other substances. Food in the form of sucrose is first loaded onto the phloem tissue by utilizing of ATP energy. This increases the osmotic pressure of the phloem tissue. As a result, water starts moving into the phloem tissue as a result because a concentration gradient has been set up due to osmosis. When water moves into the phloem tissue, there is a build-up of pressure within it. As a result now, food starts gushing down to those areas within the plant where the pressure is low. Or in other words, we can say food starts moving from the source to the sink. Let's correlate with this diagram. This is the place where food was loaded onto, and then slowly the food started moving on to those regions where food is required. Hence, it is also known as the mass flow hypothesis. Food moving from high pressure areas to low pressure areas where food is required. Now let's have a question answer session on the topic of transportation. It will be a rapid fire round, children. Let's begin. They ensure that blood does not flow backwards when the atria or the ventricle contracts. Name them.
Valves.
Good. The blood vessels that carry blood away from the heart.
Arteries.
Very good. Can you mention the two functions of lymph?
Yes, ma'am. The first one is the digestive and absorbed fats from the intestine are carried away for assimilation, and the second one is it helps in draining the extracellular fluids, that is lymph, back into blood.
Okay, very good. Can you define double circulation?
Yes, ma'am. In mammals and other vertebrates, the blood moves through the heart twice before it gets distributed to the different parts of the body. And this is called double circulation.
Good. Name the main force which drives water up the xylem vessels.
Yes, ma'am. Transpiration pull.
Define translocation.
Ma'am, the process of transport of food through phloem has been defined as translocation.
Okay. In the human body, two types of fluid help in transport. Can you name them?
Yes, ma'am. Blood and lymph.
Now let's begin our next topic of discussion, that is excretion. Children, can anyone define excretion?
No, ma'am.
Okay. It's a process by which harmful nitrogenous wastes, excess salts and excess water is thrown out of the body. Nitrogenous wastes include urea, uric acid. Now let's study the components of the human excretory system. The human excretory system comprises of a pair of bean-shaped organs called the kidneys. Here you can see blood is entering into the kidney through this blood vessel and leaving through the vein. The kidneys open up into a pair of tubular organs which are known as the ureters, one on this side and the other on this side. Both the ureters open up into a muscular organ called the urinary bladder, and the urinary bladder ultimately opens into the urethra from where the urine is thrown out from time to time. Each kidney comprises of almost 1 million tiny filters which are known as the nephrons. Here we can see the nephrons which are present within our kidneys. What's the function of the nephron? They act as filters. They help to filter the blood and separate out the nitrogenous waste products like urea, uric acid. Now first let's study the structure of the nephrons. Here each nephron comprises of a cup-shaped head-like structure which is known as the Bowman's capsule. Within the Bowman's capsule, a tuft of blood capillaries are present which is known as the glomerulus. The Bowman's capsule opens up into a long U-shaped tubule which is known as the Henle's loop. The Henle's loop ultimately opens up into a broad collecting duct, and the collecting duct ultimately drains the urine into the ureter, from where it is carried down to the bladder to be thrown out from time to time from the urethra. The diagram which has been showed here, it shows two nephrons opening their contents into the common collecting duct. One more thing, children, I'll tell you here. Each nephron, from the glomerulus, a blood capillary arises which intricately girdles the entire Henle's loop of the nephron and leaves the nephron as a branch of the renal vein. Now let's discuss the function or working of the nephron in detail. Check out, children. Blood laden with wastes like urea, uric acid, etc. first enters the glomerulus, where a very high pressure is created. Due to this high pressure which is created, the blood gets filtered and gets collected in this cup of the Bowman's capsule. The filtrate which gets collected here, children, the plasma part of the blood gets filtered. The filtrate which gets collected here, it comprises of useful substances also, like it has proteins, amino acids, glucose, excess water, etc. Plus it also carries the wastes, that is urea, uric acid. The entire filtrate having useful as well as harmful substances, this filtrate now starts its downward journey through the loop of the Henle. While this filtrate is on its downward journey through the Henle's loop, something happens. The useful substances like the amino acids, the proteins, the glucose, etc., that gets reabsorbed by the blood capillary which has intricately girdled the nephron. So the useful substances, they get removed from the filtrate. Ultimately, while the filtrate moves up the loop of Henle, and by the time it comes and reaches this collecting duct, only the excretory wastes, that is urea, uric acid in an aqueous medium, that gets collected in this collecting duct. So, two things to be noted here. While the filtrate is moving down, useful substances are being reabsorbed by the blood capillary and being returned to the body. What is being thrown out? In a very hypertonic medium, the wastes like urea, uric acid, they enter into this collecting duct. This collecting duct, children, ultimately goes and opens up into the ureter. The ureter will empty its contents into the urinary bladder, and now this filtrate gets a new name which is called urine, and it is thrown from time to time from the urethra and thrown out of the body. So this is how the nephron works within your kidneys to throw out urine, and together with it nitrogenous wastes, harmful toxic wastes like urea and uric acid are removed from our bodies. The amount of water to be reabsorbed by the nephron tubule depends upon two major factors. Let's refer. If the body undergoes water stress, the concentrated urine will be thrown out, that is, more water will be reabsorbed by the nephron and vice versa. If the amount of waste to be thrown out is more, then more water will be released as urine. Now let's talk about excretion in plants. Can you tell me how does a plant excrete its wastes? Yes, children. Now let's have a look. Here we have a diagram for us. Excretion in plants can be carried out by all these different ways. First, the excess water from the plant body is released by the process of transpiration. Gases like CO2 and oxygen, they are thrown out of the plant body by the process of diffusion. Some wastes are excreted directly into the soil by the roots of the plant. Some wastes are stored in the vacuoles of the plant leaf cells. Resins and gums, they are sometimes stored in old xylem vessels. And last but not the least, some wastes are removed by the withering of leaves. You might have noticed during the autumn season, some of the plants, they shed all their leaves. Do you know what the plants are doing? Actually, they are excreting their wastes. Whole year round, they store the wastes within their leaves and wait for autumn to come. During that time, they shed all the leaves and get rid of the wastes.
in term 1, the first experiment is to prepare a temporary mount of leaf peel to observe stomata. For this particular experiment, certain requirements are there. We can see what are the requirements. That is slides, cover slip, dropper, forceps, needle, some leaves and petri dish. This is watch glass. Then we need saffronine, which is the staining agent. We need glycerine, which is mounting agent, and water, which is kept in a beaker to wash the peel after staining. Let's see how the preparation can be done. First of all, take the bryophyllum leaf and fold it towards the back side. This is the back side of the leaf. Fold it towards the back side. When we fold it and stretch it, we have to just pull it this way. We can see a peel is coming out of the leaf. This particular peel can be taken off, and it can be, with the help of a forceps, it can be separated and kept in water immediately. We can see the peel. Keep the leftover leaf and dip it in water immediately to prevent desiccation. After that, we have to take the peel and dip it in the stain, which is saffronine. Put it for 2 minutes. We can see at the watch, or we can take it this way, and keep on moving it so that it doesn't float at the stain and takes uniform stain. Once the stain is taken, we have to take out the peel. You can see it has been colored, and then we have to remove excess stain. So wash it in water, because we have to wash it twice. So let's take the previous petri dish. In that, just shake it, and you can see the excess stain is coming out. The peel is this. Again, wash it in fresh water. Twice washing should be done so that excess stain is removed. After that, we have to place it on a slide. Precaution is to hold the slide with edges so that there are no finger marks on that. Place a slide, take the peel, normally it will curl on, and then place it on the slide. While placing, when the leaf curls, we have to remove the curls and spread it. Mounting of the material should be done in the center. So you can see the peel is there. Once it is kept, then with the help of the dropper, we can take either the mounting agent, which is glycerine. While putting glycerine, we have to hold it like this, because these are beaked lids and they should be towards the joints so that the glycerine will come out. Now put 1 drop of glycerine gently over the stained peel. As we know, glycerine is mounting agent. After that, take a cover slip and place it at 45 degrees near the glycerine. After that, with the help of needle, we can hold it like this, and gently we should leave it. This way, you can see the slide preparation is over. Always hold the slide with edges. So this is a prepared slide, which is temporary mount of the leaf peel to observe stomata. After that, focus it in low power. Numerous stomata will be visible in brighter field, and then focus it in high power. We'll see only 2 or 3 stomata with high magnification, bigger in size. In this experiment, various areas are covered while asking the multiple choice questions. For example, it can be asked that what are the requirements while preparing the slide. The requirements are in front of you. They can be listed in the multiple choice questions, or accordingly you have to tick mark them. So all the requirements are there, like slide, cover slip, dropper, needle, your forceps, then leaf peel, stain and glycerine for washing water. Next area is that what is the staining agent while preparing the leaf peel. So it is very much clear, saffronine is used to stain the plant cells. Then it can be asked that what is the mounting agent, what is its use, why we should use that. So glycerine is the mounting agent. It prevents drying of the cells and the peel can be left over moist for some time. Then the questions can be asked from the precautions point of view, that what are the precautions should be taken. Like here we can see the first precaution is over staining of the peel should not be done. It should not be kept for a longer time in the saffronine. Then oozing of glycerine should not take place. Here you can see that if we hold it like this, there is no oozing of glycerine out. So that oozing should not be done. Then while putting the cover slip, we should take care that there are no bubbles inside, and the slide should be, the mounting material should be in the center. So all these, if you do it carefully, then you can have all the answers related to the practical. One more question which is basically theoretical, it can be asked related to labeling of the diagram. Like guard cells can be asked. Inside the guard cells, nucleus, chloroplast are given. They can be asked that what are these parts, and just mark them. So this is all about the first experiment, preparation of leaf peel to observe stomata. The second experiment is to prepare, to show experimentally that carbon dioxide is released after respiration. For this, following are the requirements. We have to take a freshly prepared potassium hydroxide solution, some germinating seeds, as I have kept them in the conical flask, a small beaker, a bent tube which is U tube, then some amount of water, a small tube which has to be hanged inside it. To tie it up we need a thread, then a cork. This cork has to stop the mouth of this particular conical flask. Now with the help of the dropper, because the tube is very small, so we can take the freshly prepared solution, put it in this tube and hang it inside the conical flask. We have to cover the mouth of this conical flask, so, and it should be airtight. In this way it should be hanged. It's a cork or stopper. If the thread is too long, the students can cut it off, the extra thread, and can be kept separately. Now put some water in the beaker, and with the help of this bent tube, one end should be inside the cork, the other end it should remain immersed in the beaker. This setup, it should be kept for at least half an hour so that we can observe. The amount of germinating seeds should be more so that the respiration rate is higher. After some time we have to again observe that what has happened to the potassium hydroxide solution which is kept inside the this tube which is hanging inside, and what is the level of water in this bent tube. After that, various questions can be asked related to this particular experiment. That can be, what is the correct sequence for performing the experiment, what should be taken, then where should be the tube related to the labeling of the diagram, it means what should be the position of the distal end of the bent tube, whether it should be immersed inside of the water or it should remain outside. Then what type of seeds we should take, whether they should be germinating or dry to conduct the experiment. In the tube, which solution has to be prepared, why potassium hydroxide solution, and so on. So from these all areas the questions can be asked. We have kept this set up for some time, approximately 45 minutes we can say. Here you can observe in this hanging tube there was freshly prepared potassium hydroxide solution which has slightly turned milky. When potassium hydroxide reacts with carbon dioxide, the solution turns milky. So this proves that carbon dioxide has evolved after respiration. Due to that, some partial vacuum is created in the conical flask. Because of that, here we can observe that the level of water in this U tube is slightly raised. So this experimentally we have proved here that carbon dioxide is evolved after respiration.
The aim of our experiment is to show that light is essential for the process of photosynthesis. Now we have a large number of materials which are required to conduct our practical. Let's have a look at the entire list of the equipments and accessories that we need. First, we need a healthy potted plant, black paper strips, adhesive tape, alcohol, iodine solution, a tripod stand, we need a wire gauze, water bath, beaker, petri dish and a Bunsen burner. All these are our requirements for the practical to be done. Now the healthy potted plant which you had selected, that's going to help us out further. Take the healthy potted plant, put it inside a dark room for 72 hours, that is 3 days, 1, 2 and 3. It is de-starched. Take the potted plant out, select one of the healthy leaves and fix black paper strips firmly on both the sides of the leaf with the help of adhesive tapes. Now, next, shift the potted plant out to sunlight for a time of roughly 5 to 6 hours. Plug the leaf out, bring it to the lab and then you remove the black paper strips carefully. Now this leaf is going to be your experimental leaf in the lab. First step, boil the leaf in water to kill or destroy the plasma membrane. Next, you pick the leaf out from the boiling water. It is boiled only for about 5 to 10 minutes. Take the leaf out. Boil it in alcohol in a water bath. Alcohol always has to be kept in a water bath because it's inflammable. After boiling it in alcohol, what happens? The entire chlorophyll from the leaf is removed. The leaf gets decolorized. Fine, you can see the leaf is totally decolorized, color has changed. Pick it out and wash it in water, keeping it in a petri dish. Excess alcohol to be removed and the leaf gets softened. Pick it up from there, put it in another petri dish and now you add few drops of iodine solution onto the leaf. What do you see, children? Instantly there is a color change. Pick it out. We may even wash the leaf once again to remove the excess iodine on its surface. Have a look, children. The part of the leaf which was exposed to the sun, which received sunlight, those two segments have turned blue-black, showing the presence of starch. But the part of the leaf which we had covered with black paper strips that did not receive sunlight, as a result you can see it remains pale yellow. So there is no starch which has been formed in this part of the leaf. So this experiment proves that light is very much essential for the process of photosynthesis.